presentation. My talk is titled Human-Wildlife Interactions of Hawaiian Waterbirds in Wetlands. I'd like to thank my Capstone committee, Dr. Melissa Frank, Dr. Felipe Winter, and Dr. Travis Vidal for their help in the project, as well as all my study participants whose work made this possible, and my funders, the Hawaii and Lowell Foundation, and the Undergraduate Research Opportunities Program, as well as the Hawaii Wildlife Ecology Lab, PICO Lab, and my two research assistants, Lena and Ariel. So, begin. Human-wildlife contact. There is both a positive and a negative component for this, for humans and wildlife. They have lots of chances to interact with each other, but recent anthropogenic change has resulted in large losses of biodiversity that we typically see today. This is usually caused by climate change, habitat loss, overharvesting, as well as the introduction of invasive plants and predators. However, there are also benefits that have been recorded between humans and wildlife, such as ecotourism, social well-being, people's connection with nature, and mutually beneficial land use. Coexistence is possible, and as sea levels continue to rise, we can expect to see that the amount of available habitat for our species will continue to decline, and this will place greater emphasis on the areas of land where we have people and wildlife coming into contact. And because of that, it's important that we understand how they're interacting to better support species recovery. The different types of wetland sites in my project included agricultural wetlands. These primarily were focused on crop production and had actions such as weeding and planting. And in these types of sites, there were frequent interactions that occurred, and these sites were typically private. Within this group, there was the subgroup, the aquacultural wetlands, whose goals revolved around seafood production. And actions for this involved spawning and restocking, feeding, and harvesting. There are a little bit less interactions here, but again, this is typically a private wetland type. My next category of wetlands were recreational wetlands. These primarily focused on human leisure, and actions such as grounds maintenance, recreation, and even feeding occur here. And there are very frequent interactions between humans and wildlife here due to their public access. And finally, we have wildlife sanctuaries. So these types of sites revolve around wildlife habitat. The primary actions occurring here involve predator and plant control, monitoring and research of endangered species or protected species. And typically, we'd expect to see fewer interactions here due to the lack of public access. There's just kind of a scale for what we would expect for the amount of disturbance by wetland types. And you can see wildlife sanctuaries, typically we would expect to have the least amount of disturbance, with recreational sites on the opposite end of the spectrum with the most amount of disturbance. So, human-wildlife interactions, what influences them? It's complicated. There's a big list of factors that influence them, and I'll go through each of these bullet points with you now. So, disturbed versus non-disturbed populations. Look at the picture on the left. This is a wildlife refuge, and as you can see, there's a fence excluding people from the wildlife in the sanctuary. And on the opposite side of the slide, you can see some ducks eating bread. But you can imagine these birds behave very differently from one another, and that's due to the public access differences between these sites. Next, we have the disturbance type that that the wildlife are experiencing. On the left, you can see some coots watching around a gulf. And on the right, you can see a researcher setting up a trap for some monitoring on a wildlife sanctuary. Onto habituation. Again, people feeding birds, everyone feeds ducks. It's cute, kind of fun. But is it good or bad? We don't fully understand or know all the nuances of it. But these birds do get habituated, especially when food is involved and they know that there's some sort of benefit for coming around people. However, not all bird populations would react the same. On the opposite side, you can see what would happen if you would maybe try to feed a bird in a wildlife sanctuary. It's friendly. And next we have environmental factors. We have different types of wetlands where these wildlife are residing, different amounts of water, as well as land use goals. And finally, we have body mass and species. So we can expect that a Koa, the bigger bird, might behave differently. It might not want to fly away as much as an Io, which is a lot lighter and smaller. And that brings me to my 4 study species. So starting on the left-hand side, we have the Io, or the Hawaiian stilt, the Alaiula, the Hawaiian gallinal, the Alaike'okeo, Hawaiian coot, and finally the Koloa maoli, or the Hawaiian duck. Just as a little side note, the Koloa maoli here on Oahu are widely hybridized, but for the sake of ease, I will just be referring to them as Koloa maoli for this presentation. So, how can we know how birds feel about the stewards in their environment? One way is by looking at their behavior. So here we can see a diagram that I made, and it has a few different differences, or distances, and I'll walk you through them now. So typically, once I'm on site, I'll use a laser rangefinder, as you can see on the top, and I'll use this to measure our starting distance. So that is the distance between our pedestrian approaching our bird. That's that distance where they're first starting. We record that, as well as our alert distance, which is subsequently when the pedestrian begins approaching the bird. When this bird might change its behavior, maybe it chirps, looks up, stops what it's doing. That would count as our alert distance. And finally, we have our flight initiation distance. And this is the point at which our bird takes off, runs away, swims away, whatever it may be, but it is running away from us. For our approaches, we had 2 different groups. We had stewards and non-stewards. For our stewards, these were people who were frequently stewarding a wetland and who we thought that the birds would be familiar with. And for our non-stewards, this was either me or my field assistants. And one thing to note here is that clothing was standardized for our approaches. For our non-stewards, we wore green logo shirts, and for our stewards, they wore their typical field clothes that they'd be working in. Our first research question is, do water birds respond differently when approached by a wetland steward compared to a non-steward? And for this, I have 2 hypotheses. They were that water birds will alert at a shorter, there will be a shorter alert and a shorter flight initiation distance for water birds when approached by a wetland steward compared to a non-steward. And my next research question is, what is the relationship between distance measurements and the frequency of wetland stewardship, individuals' attitudes towards their water birds, their interactions with them, and their interest in fostering a connection with them? So my first hypothesis was that stewards with more frequent stewardship would be able to get closer to the birds. Next, stewards reporting more positive attitudes towards the birds would be able to get closer. Stewards reporting more frequent interactions with the birds would be able to get closer to them. And finally, stewards reporting more interest and connection with the birds would be able to get closer as well. You can see a map of my study sites across the island of Oahu. Data collection occurred last year between January and November. And I surveyed a total of 32 different wetland sites, each with their own unique location, management style, and perspectives towards the water birds living there. You can also see a pie chart of the breakdown for those 32 sites by the wetland site type. This brings me to my methods. See my wildlife permits here, as well as my IRB, but I will walk you through these boxes. So, beginning with my distance measurements, like I explained before, these are our alert distance and flight initiation different distances that we measured with our laser rangefinder when approaching birds. Next, I conducted point counts at each of the wetlands. These point counts consisted of a 5-minute period where within the perimeter of the wetland, all migratory shore and water birds were recorded, as well as native water birds. And I also recorded the amount of people in the wetland, as well as within a 5-meter distance to the edge of the wetland. And next we had our steward survey. Here you can see, it's not showing. You're supposed to be able to see a QR code, so you can scan it to see the specific questions that I asked, but they ask things such as how frequently do you manage water birds, do you like or dislike them, do you interact with them, and how interested in connection are you with those water birds. So with those different data types, I first ran a paired t-test between our stewards and non-stewards for how closely they were able to approach. Next, I ran multiple linear regressions using our other data types to try and match models to what we were seeing in our distance measurements. And these were the variables in our multiple linear regressions. So they were grouped into two sides. We had our environmental and our social. Starting distance was used as a covariate in both because it was shown as significant according to the literature. Environmental, we had our flock size, or how many birds of the same species are hanging out together, how many people are in the wetland, and how many of those surveyed birds are in the wetland. For our social variables, we had, again, starting distance and stewardship frequency, feelings towards birds, interaction, and level of connection, all from our steward survey. And finally, from our multiple linear regressions, if we had a model fitting score of less than 2, or AIC, it was then, we used AICC model selection for our models. And if there was a delta less than 2 for these models or no clear top model, then they were averaged. This is kind of what my data looked like. Although I did survey 32 sites, I did not get 2 surveys back, and every wetland did not have every water bird type as well. You can see the numbers broken down by species here. And due to the small sample size of Alaike'okeo, they were unfortunately excluded from the linear regressions later on in this presentation. But on to the paired t-tests. A graph here with our species on the bottom axis and our mean alert distance on the vertical one. And you can see the breakdown where pretty much the main take-home message is that stewards were able to get closer to water birds compared to our non-stewards before they alerted. And stewards are in blue, non-stewards are in red. Next, for our mean flight initiation differences, you can see the same style graph. And again, we see that stewards were able to get closer to our water birds. Stewards were able to get closer to our water birds compared to non-stewards before the birds flew off. And on to our linear regressions. So here is our model selection. We have our model name on the left, followed by our K value, which is the number of variables in our model. This does not include our intercept, just an FYI, and our AIC number. So this is just a number for how well fitting our model is to our data. And then our delta, which is it's the difference between the current row and the top. So again, for any delta less than 2, we model averaged these results. And finally, we have our weight, which is the importance of our model at explaining our data that we saw. But here for the Alaiula, we can see that number of people, total birds, flock size were important in our models. One important thing to note is that although starting distance is included in all of our models, I will not be reporting on it because it is covariate. We knew it was going to be important going into this project. And it wasn't exactly something we were interested in measuring though, but its importance was worth considering. So it's included as a control. Here we have a graph of our total bird numbers and our flight initiation distances for the Alaula. And we can see that higher total birds increase the distance before flight. So kind of confusing with the increase and decrease for our distances, but basically a higher distance, I think it's like of a longer distance, they weren't able to get as close. So more birds, further distances before they were flying off. Oh, and for the sake of time too, I'll only be showing you our flight initiation distance models and not showing our flight distance. Onto our social models, we saw that connection was pretty much the top model at explaining the data that we saw. There wasn't exactly a clear top model with starting distance as their second best, but even considering that, these first two models did account for almost 50% of the cumulative weight being seen here. On to our I/O for our environmental regressions. Again, we can see our models and what variables are included in there, our K column, our AIC ranking, the delta difference, as well as our weight. But we can see that flock size and number of people were the best explainers for our numbers we were seeing. And again, starting distance alone is kind of our null model, and with that as our top, kind of shows that, I'll get into it in the discussion. And next, for our social model, we just had one that was our top performer that included connection and feeling. This model, what was interesting was that it accounted for over 50% of the model weight for what we were seeing, and a pretty high number. So must have been onto something there. And here we have our plots. So this is for interest in connection with stewards and the flight initiation distance of our I/O. But you can see that most people put 4s and 5s. And what was interesting here was that there was a significant difference between 4 and 5s with, we can see a little bit of a, kind of due to the small sample sizes, and a little bit of a flat line with our neutral option from our survey. But towards the other end of the survey, stewards were able to get closer before the birds flew off. Next, for a level of feeling, we saw that more positive feelings increased distance before flight. So, more you like the bird, kind of a weird, weird finding, but the more you like the bird, the farther it flew away from you. We'll get into that later. The breakdown, survey options. Next, for our last I/O model, we can see weekly stewardship frequency and flight initiation distance. And there we can see a big weight on the extensive option from our survey, as well as a almost significant finding here, very close. I'll get into it a little bit later. Onto our Koa Maoli. When looking at our environmental variables, we saw that number of people was our top model. None of the other models came close to explaining the data that we were seeing. And here is a plot of the number of people and flight initiation distance. And we can see that as the number of people increase, you're able to get closer to the Koa Maui before they fly off, however we're expecting. Next, onto the social side of things, we can see that connection and interaction are the best predictors for the distance measurements we were seeing. There was a big heavy weight, cumulative weight here, which means we were hopefully onto something with this. There's a plot of that. We can see our interesting connection on the bottom axis and our flight initiation distance on the vertical axis. And you can see the distribution of Stewart's answers in relation to that. Not significant, very close, but still interesting. We can see the interaction frequency of Stewart's with the Koa Maui in relation to the flight initiation distances that we were recording. We can kind of see that around the middle of the survey answers with neutral, and sometimes our flight initiation distances kind of were similar. And on the ends of the spectrum, they dropped a bunch. So, what is this all?
In the interest of time for everyone to go, I'm going to have you up here. Appreciate it. We'll have time for a question if folks have. Obviously, we're going to get to this discussion, so if there's a couple of burning questions, we can take one of those now.
Hello, my name is Patricia, and this project was in partnership with Honolulu Fish Pond Heritage Center, conducting MEM surveys at Kalauha'iha'i and Niu to inform ecological restoration. My capstone panel is Dr. Litton, Dr. Travis Ido, and Chris Kramer. So on a global scale, limu that are native to the region have been evolving in place for millions, and for some species over 1 billion years, creating an optimal habitat for a diversity of native organisms. As primary producers, they form the foundation of these complex food webs, ultimately shaping the structure and function of our nearshore environment. They're also an integral component to our nearshore carbon cycle, and they stabilize coastal structure by breaking up wave action and stabilizing coastal sediment. Limu in Hawaii has been evolving closely alongside the influence of fresh water, above ground, but notably below ground fresh water that delivers at a natural pace into the ocean. This delivery is called submarine groundwater discharge, and it creates a gradient in our nearshore coastline that lowers salinity and delivers key nutrients for a diversity of species to flourish. Uncle Wally let me know that Uncle Henry could stand at the coastline, and by what limu was present, he could tell you how well people were caring for the fresh water from the top of the mountain all the way into the ocean. Limu is also very culturally significant to Hawaii. So Hawaiians identify limu as one of the first species to inhabit the earth. Therefore, they explored its sacred purposes beyond its ecological functions, using it in ceremonies of ho'oponopono, which is a reconciliation ceremony to set things right amongst family members or community members. It's also found in mo'olelo, or stories that are passed down through generations, informing people of today about what's going on in our world, you know. And it's also used as an adornment in hula. It's used for its rich nutrients in la'au lapa'au, and also as a food staple alongside fish and poi. However, in modern day, the abundance of limu has decreased so severely that it's now more of a condiment that's kind of hard to come by now. The loss of limu in Hawaii has been happening due to urbanization, altering the landscapes and hydrology that blocks the natural flow of fresh water to the ocean. It's also due to harmful land use practices, which creates this stormwater runoff and a high amount of nutrients into our coastline. Climate change, the introduction of invasive species, and destructive harvesting practices. So with all that said, I'm going to take us to where I conducted these surveys out in Kalauha'iha'i in the ili of Niu on the island of Oahu. Kalauha'iha'i directly translates to break or the shedding of leaves, referencing this large amounts of volume of water that would come from the uplands of the mountains, in artesian spray from the uplands of the mountains and flow out through this fish pond and out to the bay, causing a break in the reef. It's also referencing the shedding of the Aikapu system, which was the natural resource management system that Hawaiians used to manage their resources. This was a place of abundance and the summer home of King Kamehameha and Queen Kahumanu. Freshwater here was so abundant that horses could drink out of the bay. Mrs. Norma Correa, a longtime resident, would talk about how when she was a young girl, she would go out and with her friends and collect species like limu manawea, limu vavaiole, limu kohu, and limu palahalaha. And they would bring it back into the shoreline and all gather around and help clean it and help prepare it to take home, showing that limu is not only so important for our environment, but it also extends into these relationships with each other. These are some of the common native species that occurred during when I was doing my survey. We have some reds, greens, and browns in here. So back to Kalahaiha'i, fast forward to the 1990s, this spring water was blocked due to a construction project which widened Kalaniana Ole Highway and adjacent highway there. The spring water was blocked and then diverted to the sewer system, which is still happening today. This ended one of the last two Hawaiian fish ponds along this bay. However, Mauna Loa Fish Pond Heritage Center has been working with area legislators and community groups to release this water back into the bay, which is expected to help with native habitat. So in preparation for this, I conducted a baseline study of what limu is out there and environmental variables associated with this distribution to get a really good understanding of what the composition of this area is in particular. So I assessed limu and environmental variable distribution at Kalahaiha'i. I examined the relationship between limu and environmental variables, and then I made management recommendations for post-spring release. So this is the study site. This area is mostly made up of pavement. So pavement, not as in construction pavement. Pavement is a term used to describe a flat foundation of dead reef, kind of thing. And then there's reef rubble, small pockets of reef rubble, sandy depressions, as you can see with the lighter blue colors, and then aggregate reef out around the breaks that supports marine life and has like coral substrate and nice solid rock life. So these are the transects that I laid out. I laid out 17 20-meter transects from the shoreline out to the reef crest. And at each transect, I sampled macroalgae and environmental variables. So with the limu collection, I used the line point intercept sampling method where I had a meter stick and marked 3 even locations on the meter stick. And then I laid it at each transect, and at each meter, I did 3 pinches. Something to note is that I recorded this information based off of its presence, not its abundance. So if I pulled up a whole lot of Acanthophorus crucifera, but a single blade of Palamida, I recorded it 1-to-1, but I was still able to see the frequency in which they occurred across the site. And then I sampled environmental data, which included the temperature, salinity, dissolved oxygen, pH, nitrate, and turbidity using the YSI. And I sampled that 5 times at each transect. Also, something to note is that I grouped my data by rows to see the gradient. So when we see the charts later, row 1 is at the shoreline and row 5 is out toward the reef crest. So Malama Mauna Lua is also another community group that has been doing work out here since 2009, doing limu huki days, removing invasive species and repurposing it for compost. Their removal sites have extended toward the row 4, but they are currently focusing on the areas of rows 1 and 3. Their monitoring methods include plots, quadrats, and percent cover. So the analysis I used was I visually plotted environmental variables from the shoreline to the reef crest. Then I visually plotted species presence and frequency from the shoreline toward the reef crest and then separated native and non-native. And then I used the Pearson correlation coefficient to assess relationships between the limu and environmental variables. So this is the graph showing my environmental variables, and just to kind of like get us oriented, row on the y-axis we have the rows. So row 1 we have shoreline, row 5 we have reef crest, and the 0 on the y-axis is the mean for each environmental variable that is marked. So the environmental variables are the colors, are marked by colors, and so those are the means for each one at 0. And if anything above the mean, if the bar goes above the mean, that means it's elevated, and then if it's below the mean, that means it's less than the average. And anything outside of 1 and -1 would be outside the norm. So, so as you can see, at the shoreline, there is a lot of influence by human influence here. So like there's warmer water, well, not human influence, but the shoreline has warmer water, lower salinity, and the nitrate came in at around 3.9 to 4.7 micromoles, which is slightly elevated, but it's nowhere near pollution. It shouldn't be anything to worry about, you know. And so as we look at the nitrate, which is shown with the orange bar, it starts off elevated towards onshore and then it decreases offshore, which is kind of a natural decrease, a natural progression for nitrate, you know, because we move a little bit further away from human influence. And then turbidity also improves offshore, so the water was a little less murky. As you can see with the gray bar, turbidity is very high near shore with all that sedimentation, and then it decreases offshore. And salinity also increases offshore. It gets a little saltier like the further you go out. Showing that row 5 is more ocean influenced cooler water mixtures. So species, this is the species composition of the bay. So again, we got row 1 through 5, row 1 being closest to shore. And then so the red bars are the invasive species and the colorful bars are the native species. And here we see that there's overall species, with species present, there's less invasive species. And I think likely in rows 1 and 3, this is likely due to Malama Manarua's Liuhuki days. Row 4 and 5 is a little bit more natural environment, so it kind of takes care of itself a little bit. And also another thing to note is that there are more native species counted than invasive species. So starting with invasive species, we have 3. There are Acanthophorus spicifera, Avernveria lacerata, and Gracilaria salicornia. So Gracilaria salicornia and Avernveria lacerata declines as you go offshore, suggesting that they like the more lower salinity, higher nutrient environments. And Acanthophorus spicifera dominates the whole site, showing that this whole site is great for them. Native species that decrease offshore. So for example, Limu hulupua'a, which is Spiroidia filamentosa, it decreases offshore. These are all species that decrease offshore, showing that they like the higher nutrients, lower salinity levels. So this suggests that post-spring release, this may increase habitat for them. You know, maybe pushing other species out of the way, but it also might be create more habitat for like our manaweas, you know, which would be a good thing. And then these are native species that increase offshore. So these are species that like low nutrients and higher salinity, suggesting that post-spring release, this may push species further out. So while it may push species further out, it might also create, the spring release might also create more habitat for limu to occur. So this is the correlation table that kind of brings everything together. We have the species on the x-axis, the environmental variables on the y-axis, and the red boxes are a positive correlation. So if the environmental variable like nitrate goes up, the species with the red box will go up too, the abundance will go up. So, and if the blue box is a negative correlation, so if the environmental variable goes up, the reuse abundance will go down. And this kind of just brought everything that we just talked about together, that Gracilaria prefers lower salinity and higher nutrients, Alameda discoida prefers higher salinity found offshore. In conclusion, spring water return may create a water quality gradient. Alameda discoida may be pushed further offshore post-spring release, and Acanthopus chrysoptera may respond to this gradient, offering insight into what they prefer. What is their preferred habitat? And then this may also create more habitat for Gracilaria species, so improved conditions for our native Gracilaria parvispora and cornucopolia, and they were found in rows 1 and 2, so I would suggest outplanting in rows 1 and 2 around rocky substrates, kind of avoid the sediment areas, and then continued removal for Gracilaria salicornia. Next steps would include conduct a post-spring release survey, map the freshwater plume entering the bay, and then conduct a herbivore count, because those are kind of like the unseen grazers that that kind of create different differences in what we find out there. And then apply these findings to freshwater advocacy to show before and afters. Thank you to everyone who has supported me throughout this program. I truly appreciate it.